I'm Deborah Grady. I'm professor of medicine here at UC South and director of and I'm going to talk this morning a little bit about blinding interventions and controls in clinical trials. So let me ask you all before we get going, how many of you think you might actually do some sort of trial? Or some sort of raise time? Oh, wow, okay, great. All right, and for the rest of you, you may think this is not entirely pertinent, but I actually think it is, because as you go through designing observational studies, a lot of the principles we talk about you can't entirely do. That is, you don't have an intervention perhaps, but you're thinking about creating one, right? You're thinking, if you're thinking about an association between a risk factor and an outcome, you're also thinking about some way perhaps to modify that risk factor and improve the outcome. And the other thing is that we really do kind of think of clinical trials as the best clinical research design. It's clear you can't always do a clinical trial, but often what you can do in your observational studies is to use some of the principles that we'll talk about. So for example, today when we talk about blinding, observational studies aren't blinding, of course, but so hopefully a lot of this is also pertinent to just good study design in general. So we're going to talk a little bit about why blind, what it is, and the main problem, of course, is what to do when it's just difficult or impossible to blind an intervention. So somebody answer me. If you can't answer this question, you have to leave. Why randomize?
Limit.
It's fun, it's easy.
To minimize confounding.
To minimize confounding, but how do you say that in English?
Make your groups equal.
Right, so both of you said to make your groups equal. So, the groups are balanced when?
At the start.
It's the day you do it, right? So it balances, and here's the other important thing, and this is the really important thing about randomization actually is, in observational studies, you can measure age, ethnicity, body mass index, whatever. If those are different between the groups, you can use elegant statistical approaches to balance those up using models. And that really works. That's why observational studies tend to give us good answers to questions. The problem is there are often confounders or differences between the groups that you don't know anything about, in which case you can't measure them. Or maybe you do know something about them, but they're hard to measure. And if you can't measure, if you don't know what they are and you can't measure them, then you can't model them, you can't use statistics, and you can't balance them up. So it balances both the measured and the unmeasured variables. Now here's the important thing is, it does that at baseline, right? It does that on the day you randomize them. And these are like people, they're not rats or mice. So after the day you randomize them, all sorts of things can happen that then make those groups no longer very common, even if they started out pretty common at baseline. And that is the value, mainly, of blind. So why blind? Well, your effort here is to maintain the balance of these groups throughout the whole intervention. And that's what blinding is really all about. How does it do that? Think about how it does that. Well, really, the idea here is to eliminate these three things. And again, this is stuff you might think about even if you are not doing a blinded trial. It eliminates co-intervention. We'll talk about that in a minute. So interventions that aren't the one you're interested in. It eliminates biased outcome ascertainment and biased measurement. I'll just talk a little bit more about those things. So I'll just give you a couple examples. Here's an example. The women's health study is close to 40,000 women healthcare providers. They were randomly assigned to take low-dose aspirin every other day or placebo. And the follow-up was 10 years. The outcomes were cardiovascular events and deaths. It's a pretty standard cardiovascular intervention trial testing the effects of aspirin on coronary outcomes. What could be the co-interventions in this kind of trial? So here are women, they know they're in a trial, right? Of course, you can't blind them to knowing they're in a trial. You can blind them to knowing whether they're on aspirin or placebo aspirin. But what if it wasn't blinded? And what if you're one of these women, you get randomized to aspirin, where you get randomized to a sugar pill and you know it, or you get randomized to nothing, let's say, usual care. Usual care would be that. Do you think the study staff, are going to treat the participants the same? Yeah, usually, study staff are enthusiastic about their trial question, right? They believe in aspirin. So if they know that they know some of the participants are on usual care, not on aspirin, they may be a lot more inclined to say, better get your exercise, better follow your diet, better take your statins, better stop smoking. So all of those things are co-interventions, right? And we know that those things could be effective. So these are unintended, effective interventions. And so that the participants use other therapies or change their behavior. And it's not just the study staff, but, the participant's doctor, the participant's family. The participant's family says, oh, you're in this trial, you're getting usual care, you better, exercise, follow your diet, take your medicines, etc. So this is co-intervention. Now this probably happens in all trials, right? Like, let's say this really was blinded. Because the study staff who run this usually who are they? They're cardiovascular nurses and stuff, right? They're really into preventing cardiovascular disease. There's often co-interventions anyway, but at least it would be non-differential to both groups. They'd be telling everybody, they'd be giving everybody the same advice about, diet and stopping smoking and so on. That may decrease your power if it decreases the overall event rate of the trial, but at least if it's non-differential, it won't cause bias. But if it's differential, and the differential part is what comes in because of unblinding, then it really can cause bias. And it's particularly bad if the co-interventions are happening in your intervention group. And that can happen too. You can see how that would happen, right? Because, again, somebody's very enthusiastic about whatever your intervention is. Let's say it's a new behavioral modification. They really want it to work, and they just not only want people to have these outcomes, so they just really try to give them all the advice and help they can. So that's co-intervention. Here's another example. This is an old trial, really old trial, of about 18,000 women of reproductive age who were randomly assigned to take oral contraceptive pills for their usual birth control method. They were followed every 6 months for 2 years to look at pregnancy rates, and of course pregnancy rates were much lower in the OCT group, but venous thromboembolic events, such as blood clots, particularly in the lower legs, the risk of that was increased about 5-fold. Now we've known for a long time that estrogens and progestins in the birth control pills can increase blood clot risk. So what's the problem here? This is an unblinded trial, right? And every time the women who are taking birth control pills come in and they've got at least a little swelling in their leg, the doctors go, yikes, and send them off for an ultrasound of the leg where, lo and behold, they have a blood clot. Well, lots of us are walking around with blood clots. And the women in the usual care group, they come in and they got a little bit of a swollen leg, and you say, oh, you did too much running yesterday. And that's differential outcome ascertainment. It's different from measurement. It's ascertainment. It usually does lead to differential measurement, because in many of these trials, the measurement, the diagnosis of blood clot was an ultrasound of the leg to show the clots there. But of course, you didn't get the ultrasound of the leg unless somebody thought you needed one, because they didn't, you don't go around ultrasounding 18,000 women. So this is biased outcome ascertainment. So if you know what the group assignment is, then the participants themselves, so let's say the participant knows that they're on some brand new immunosuppressive drug, rheumatoid arthritis trial or something, and they'd be much more likely to complain of all kinds of symptoms than if they knew they were on an inactive placebo or they weren't on anything. So the participants may report symptoms more. The study staff may elicit symptoms more and take them more seriously.